Okay, well, welcome back. Thank you for coming. This is lecture 8 for our Dynamics on Networks class. As usual, I need to remind you that the class is live streamed on YouTube and available publicly afterwards. And today, the plan is that we're going to hear a little bit about your plans. I know we had one presentation last week, but some brief updates on your plans for your projects. And then I want to come back. We rushed through the discussion of questions we could answer. People did a homework problem on that before we had really talked about it in class. And so I'd like to come back to that. And then I want to cover events, which people also used a little bit in class homework. And then talk about feed-forward networks a bit. And then I hope do some review of some of the homework problems. So maybe we should start with project plans. Do people have present things that they'd like to discuss about what their plans are for today?
Eden and I can go ahead and give an update on our plan.
Okay, great.
Let me just go over to the screen so I can share it. Apologies for the number of tabs in advance. Okay, can everyone see my screen?
Mm-hmm.
Okay, so after talking to Professor Glazier a couple of weeks ago, Eden and I went ahead and started with writing code to generate an arbitrary size chemical reaction system where you can specify the number of boundary species, the number of reactions. It won't always return a network exact to the specifications, especially if you ask for something that doesn't make sense. And we also made it so it was mass conserving. So that's what we're starting with. And what we have is that we have a general objective and a contingencies plan. So I'll give a high overview there, and then I'll let Eden take over to go through some of the details. Our primary objective is to play with something called chemical organization theory, which is something our advisor has been interested in and strongly connected to concepts of self-maintaining and autocatalytic sets. Our primary project pipeline is to write code that looks for networks that meet those specifications out of the random ones we can generate, and then write a series of code snippets that show how that works. And then separate of that, we would like to try to do some evolutionary runs for a specific task, but that would be probably too slow to be a good tool for NanoHub. So the idea that we were discussing was have everything that could be done in a reasonable time as a NanoHub application, and then link to, if we get through the evolutionary code, link to that on GitHub so the person could then run it if they wanted to. And then our contingency plan is that if we get a certain duration into the semester and it ends up that this stuff really isn't working out, we will turn our network generating code into an educational tool for generating and analyzing arbitrary networks on NanoHub. So that's the overview. I'm going to mute and let Eden take over.
Sure. So a big part of this is also turning chemical organization theory into something accessible through this pipeline, I think is a big part of it. It's pretty dense stuff with a lot of internal reference of language. And I think visualizations are something that could certainly be helping me as I'm trying to go through it. And I think that's something we're hoping to provide. But in the meantime, we sort of have broken down most weeks remaining in the semester into some kind of plan. We're running a little shorter on time because Connor's going to be going to a conference about 5 or 6 days before the end of the semester. So hopefully we will be done before then. So, right, I mean, we can scroll through this, but part of it is just Connor and I have got to completely finish the paper. And we want to make some pseudocode for boundary species fluctuation, as that's going to be important for our analyses, and also some pseudocode just for the assessments according to the theory. That goes well with the networks we've generated. I mean, it's got to work with Antimony. And we also want to turn that into a Python class, the generator in general, and that'll sort of solidify the backup plan a bit more. And it's something that would be nice to do anyway. Then we're hoping to implement that pseudocode we wrote the week before, try to actually flesh it out, and then get it all in one place, right? So we have one notebook that can both generate a network and analyze it. Following that, we're going to be getting ready for the mid-semester presentation, and hopefully write up the, like, have that script we got the week before presentable and sort of better documented, and anyone should be able to approach it, kind of thing. Then we'll get some feedback. This will be probably the pivot point, determining if things are working or not. And from there, we're going to prioritize finishing the NanoHub app first, like Connor said. So we're really going to try to have a polished draft that we can share and get feedback on. So in the first couple of weeks of November, we can try to have the app done, and then we can go to the evolutionary algorithm side of things in the following couple of weeks. And our plan obviously gets a little vaguer as we go deeper, but that's a lot of that has to do with just at that point, the specifics of the tasks get a little hairy. And then we have some final dates there. Yeah. So that's where we're at right now. And we're feeling pretty good about it.
I mean, I think a code generator of that kind could be very interesting to have on NanoHub, whether or not, whatever you do with it, it could be a good learning tool.
Yeah, so another option, even, I mean, we're talking about it as a backup for the class project, but I see no reason why, even if we have that as the back end, why we couldn't upload a second NanoHub app outside of the confines of the class. Because I mean, it's fun to play around with, just be able to generate an arbitrarily sized chemistry and look at the time series. So,
Especially because the rest of what we're doing is contingent on having that in the first place.
Yeah.
So.
I mean, the other thing to remember, we don't typically cover it in this class, is in principle, NanoHub lets you ask for more compute resources. You could have 4 cores if it's OpenMP by default. But if you need bigger resources, in principle, you have access to some HPC facilities. I don't know how you ask for that. And I know that setting up the apps is a lot more complicated if you ask for that.
So,
but it is possible to say you want to run on one of the high performance computing backends.
another option, I mean, my tendency in life is to bite off more than I can chew and then have to reckon with the consequences. But assuming that this is an exception and that it ends up that the evolutionary task is actually doable in the amount of time that we're looking at, something that could always be done is that even if we link to it from the outside, we could run it locally and include the results of the networks that are evolved in the NanoHub application, which might be a nice middle ground. I mean, I see Giuliano's eyes going a little wide at the requesting more resources, I think. And if it phases Giuliano, I don't know how I should feel about it.
Yeah, I know how to get apps up on NanoHub. I don't even know how to ask for more resources to be allocated to the apps.
I mean, I think let's put it this way: it's not something that happens quickly. It is possible, but I would suggest that, suppose that you could write the app so it could run, I don't know, 20 or 100 generations of evolution in a reasonable amount of time, and then you discover that you need to run 1,000 or 10,000. Then you could say, okay, version 2 of the app, if you decide to go there and you want to do it not for the course but for publication, you could try to see about asking for high performance resources for that.
Yep. And I guess you could put the app up and then be like, wait, hey, we need more resources on this thing after it's up.
Well, but you could have an app that's interesting for that runs 100 generations.
Yep.
and then say, well, you want to do a high performance app where people can run 100,000 generations. Or you could run a networks with 50 connections, but you want to run 50,000 connections. Those are all possible. So, great. Singapore sounds nice in the middle of the winter.
I'm very excited.
Okay. Okay, who would like to present next? I guess, Joel, you guys already talked. You need to talk again, or you're okay? Okay, who else?
I was going to say, I think I could go, if, especially since we're also talking about Komarova.
Okay, go ahead.
All right, here, I'll just share my screen here. All right, so Zach and I started off really ambitious here at the beginning of the semester, and we were like, oh, we're gonna, design controllers for evolutionary robots and all that kind of stuff, because that's what our experience was in. And then, as we started to get along, we were like, okay, maybe we'll dial that down just a bit, because, we were looking for something to kind of meet in the middle on, and I have this interest in regeneration in the olfactory epithelium, and Zach is very interested in development and in robotics and a lot of that stuff. So we were like, all right, let's shake hands and let's meet in the middle somewhere. And then, when we were on our other meeting with James 2 weeks ago, I think now, we were like, okay, stepping way back, and we're going to go, okay, so now we're not going to do robotics. We're going to do just regeneration in the olfactory epithelium. Connor's laughing because he's seen this all go spiral out of control, but not actually. But so we were just like, all right, so we found this Lander paper, and and we talked about that. But then actually, James had guided us more towards this other paper called that actually Joel mentioned, and which you've seen Joel and Sean talk about earlier. It's this Komarova paper, and it's this idea that cell lineages and the regeneration of cell lineages and stuff like that. So here, I'll just switch through. So one of our first stages. So what I want to distinguish very, very strongly is that we're doing something very different from what Joel and Sean are going to do. We're basically just going to be focusing on replicating the model, and also just to get very, very comfortable with just the 3 compartment systems. We're not going to go into anything more. We're just going to say maybe we'll start off maybe with 2. We'll go off into maybe 3. But especially since we're, we come from the kind of background of Randy Beer and minimal, everything, right? And we want to get a very, very deep understanding of the dynamics that underlie these systems, right? So the less kind of dimensions you have with that, the better, or the more you can try and understand. So what we're really, most of this project is really what I have on the screen right here, except more with, we're just trying to, eventually, well, I'm getting ahead of myself here. So first, the phase 1, October, what we're going to say from October 10th to 25th is we're just going to focus on implementing the model or some of the very basic. Joel has been a huge help. I had a great conversation with him the other day. He got me set up with some other things, and I'll show us something. Yeah. Anyway, Joel, super huge shout out and thank you to you. So yeah, the goal is just to create this into a application for Tellurium. At this stage from 10th to the 25th, we're going to just try and get a general sense of the narrative that we're going to talk about in the application. And we're going to start to think about what kind of analyses that we want to do. Whether or not we really want to search for limit cycles, or whether we want to just focus on finding the equilibrium points in these different topologies, or that's what we're going to start to think about. The next step is we're going to start to vary the constraining function. So what I haven't really mentioned, but I think Joel's actually mentioned a little bit, is that there's these constraint functions that the different topologies follow. Komarova goes into depth for 2 compartment systems and a little bit more for the 3 compartment systems. But what we were thinking is that possibly if we really do have this replication already, it's very bold of us to say by October 25th, we're going to have a whole replication done. But, if it gets that way, we're kind of implementing a little slack here, because it's very possible that we will not get that done by that time. But if it's done, then we can start to think about how can we start to change these constraining functions to see how they impact the dynamics of the system. And this is when we're going to really start to sink our teeth into that dynamical analysis. I've already done a little bit. Joel actually was super nice enough to give me one of the the steady state equations for one of the network topologies, and I was doing some preliminary analysis in Mathematica. So I put these equations into Mathematica, and I was doing my whole dynamical systems analysis, and I got these really wonderful trajectories here. And what you see plotted is we see S, which is the stem cells, and then what we've got on the y-axis is, right, that's Y. 3D plots always confuse me. But the y-axis, it's not A, it's actually, so that one, it was just, Mathematica was screaming at me because T is usually used for time, and it was like, no way are you putting T in us. So I just put A. Then we got D on the other axis, which is differentiated cells. So then in the paper, we're going to start to think about our mid-semester report and presentation, since we've got that on November 1st. And then we're going to start to plan out the results and discussion sections. So we're around this time, I mean, you know, this is, again, we're covering a lot, and I should actually probably switch it over to Zach here in a second. But, you know, we're going to start to think about what more we want to do. We're going to start to think about settling in and trying to wrap this into an application so that we can actually show it to people, and it's not just chaotic. The way I usually think about things is, or the way I've been thinking about things, is doing them in Mathematica first, and then, you know, kind of then, you know, scaling it back and trying to put it into something that's, you know, very much more like user-friendly in Tellurium. But that's at least the approach. That's the route that I'm kind of thinking. That's where I usually go. But yeah, then we can think about fun things like how can we implement sliders to kind of change the, you know, maybe something about the constraining functions, or maybe something about, you know, we can take a look at, like, you know, where you are in the phase portrait, or not in the phase portrait, but where you are in time or something like that. But yeah, Zach, do you have anything to say? I can switch it over here. We're getting to the end here, though.
Yeah, I mean, well, we're basically almost done. So I guess I could just wrap it up for you.
Yeah, sorry about
yeah.
that.
Yeah, no, you're fine. So phase 4, we would just basically be cleaning up everything and putting it into a NanoHub application. Oh, and we skipped to phase 5. Okay. Yeah. So, and then just basically we just draft up everything, get ready, and plan the final presentation. So this is really, by the time November 14th rolls around, ideally, you know, according to our current ambitious plan, we would basically be ready to, you know, present and submit. So then going on to phase 5, yeah. So phase 5 would just take up, you know, the rest of the semester. And that would just be submitting the actual code to NanoHub, submitting the final paper, and then of course presenting. So that's yeah.
And then wrapping it all up into GitHub too, because Zach's a huge GitHub fan.
Yes, yeah, I'm enforcing GitHub usage for our project, so that is also an aspect.
Okay, well, in 2 weeks, Giuliano will be teaching you a lecture on how to upload. You know, I was looking for the one from last year. I don't think I have the recording of your lecture on uploading from last year.
Was that the one from the
CompuCell
CompuCell class? Sorry.
No, this is the NanoHub Tellurium one.
Yeah.
Oh.
Slightly different. It's actually
It's a
easier
little
for
easier. It's a
Tellurium.
lot easier.
A lot
Oh,
easier.
okay, that's nice.
And I was going through the lectures. It was lecture 9 last semester. I don't have it.
Yep.
So I need a copy. If you have it, I need a copy so I can put it up so people can.
Yeah, I can try to find one, but
I mean, I understand that it's a little bit of a, you know, you've had to make some compromises about the level of ambition of the project, but I hope it'll still be a good learning experience for you, even if it's a little bit less ambitious.
Yeah, absolutely. No, I mean, I'm speaking for myself. Zach can speak for himself, but I'm super excited. I can't say, Zach, are you okay? Are you sad that we're not doing robots?
No, it's so I know you pushed the robot idea on there, but I mean a lot of my interests focus on neural development, and so you know, an important part of neural development is cell lineages. I mean, that's how we keep track of what neurons are where and how they end up where they are, and especially with like work like what I tried doing with C. elegans, that those cell lineages are extremely well understood. And so, yeah, no, I'm actually pretty excited. And especially, I think what will be particularly interesting is the dynamical systems analysis on it. And I think that's really where this project is going to hit its stride.
Yeah, so you may want to think about then, I mean, I think the networks that Komarova looks at almost all reach steady states or blow up. And so you might want to add one kind of feedback to them that gives you a possibility of oscillatory networks too, that you have a little more dynamics. But do the base work, and then if you have time, you could do the extension. I was thinking, boy, you could, you know, in some ideal world, you could combine with Connor and company to have him generate, you know, arbitrary numbers of components and arbitrarily connected control networks. And you do the analysis side of it. They could do the generator side of it.
We just form a gigantic supergroup.
I actually wouldn't mind. I don't know about as a this semester project, but that does sound fun. If people would be interested in doing that, I think that would actually be a fun little endeavor.
I mean,
that sounds fun.
I was at this conference last week in Vancouver, and they're trying to launch an initiative to do what I'd call digital twins of organoid culture systems. And of course, this issue of cell regulation differentiation are very important there. They had a lot of experimentalists doing pathway mapping, experimentalists doing organoid chip cultures and so on. But they also had Leah Keshen, who is a very, very famous computational mathematical biologist at the University of British Columbia. And she was talking about, she wonder if she could partner people in her class this semester with people in our class. Maybe a little late. You guys are a little too far along for that this year. But they were interested in trying to develop connections. So, but whether it happens this semester or not, there's a lot of interest in these kinds of problems. I think more, I mean, just the way these control networks get super interesting when you put them in a robot so they can interact, the robots can interact with each other in an environment. In the same way, the control systems for the cells become more interesting when you put them inside cells and then put them in an environment. So the spring course, where you do agent-based modeling, you can take these networks and then see how they play out in space. And in a sense, it becomes a little more interesting then. But I think this is fine. And again, from the point of view, of course, I understand the demands of the course are fairly modest. And so doing a good presentation of that paper, it does seem to be a really elegant paper. We keep coming back to it in different ways. And so I think making it available to a broad audience would be really great. I was also thinking, since you're talking about Mathematica, maybe you're too far along. Did you ever think about doing Stephen's summer school? Wolfram Summer School?
Oh, I didn't know that.
Yeah, I wasn't aware of that. Is that online?
I don't know. It used to be in person. I had a couple of students who took the class over the years. He's a character, of course, but he teaches himself, at least he used to. And so if you like his style, his course is a great opportunity to work with him directly. So I don't know if he's doing it this year, but you might take a look at whether Alpha has a summer school, and if he's teaching it, whether it's interesting. Because he's certainly very interested in dynamical systems. He's certainly very interested in pattern formation, and of course the application of Mathematica to do those kinds of analyses, and education. So if you could say, hey, we've combined Mathematica with this other method to do educational work, that might appeal to him. You know, Wolfram was Geoffrey Fox's. Geoffrey Fox, of course, has left now. the founder of our department of ISC was Jeffrey Fox, who now has moved to the University of Virginia. But Wolfram was his PhD student. So,
Whoa, didn't know that.
yeah, not here at IU, at Caltech. Anyway, so there are these funny connections, not really relevant, but sometimes the personal things are used. Wonderful. Okay, who would like to go next? I hear stunning silence.
I think me and Lindsay can go next.
Okay, thank you.
Yeah. So I'll share the screen, I guess, since I have everything pulled up. I don't know if you do, Grant, but might as well. Okay, so just as a brief reminder, our project has to do with activity-dependent homeostatic plasticity mechanisms, where if a neuron is too active chronically, there's an influx of calcium, and this influx of calcium somehow causes differential channel expression, changing the proportions of ion channels expressed on the membrane and potentially correcting the overexcitability of that neuron over time. So it's based, or at least the first part is based off of this paper here, where they just did a really simple bare bones model of what this process could look like. And so our first step is to sort of replicate this model, which has, again, a lot of simplifications, especially in terms of the fact that these ion channels are not at all realistic to how real ion channels and neurons behave. And so the time series of the voltage of this neuron would not look at all like a neuron, but it's a place to start. So their main finding here was that if the proportion in which calcium activates the different ion channels causes the system to end up distributed or in parameter space along these different lines, which doesn't really make sense to me based on the work that I have done about degeneracies that exist in this parameter space. I think that these really are not separate lines, but just slices of perhaps a plane of high fitness that sort of cuts through here. So the first experiment that we are going to do is to, implement these differential equations. And then instead of starting from a cloud of initial points in this little corner, start from initial points all over this volume, which I don't know why they didn't do that, but we're going to, and we're going to see if the same results come out of that. In terms of where we are now, we've been doing things in collaboratory because I had just assumed that GitHub would be hard to use with Tellurium. I guess maybe I was wrong about that. I will find out. But I knew that I could get it to work in collaboratory. So this is where we have been working. And we have everything working right with, the parameters and stuff that the paper had. And things seem to be replicating. Like, these are a couple of the time series that we have produced that look vaguely more or less like the ones that we see in that figure that I showed, except for this set of taus, which exhibits oscillatory behavior, where we don't see that in the paper, but we'll look into that. But finally, what Grant has just done is produced this figure showing that from this cloud of initial conditions in blue, which are, again, much more spread out than they were in the paper, you get these red dots after homeostatic plasticity has acted upon them, which we need to do some analysis on. but it looks like they seem to be distributed in some sort of a plane-like structure. So that's promising.
So question, this is probably something you could do fairly quickly. If I just start with one initial point and plot the trajectory in that space, what does it look like? Is it a straight line, or do you see some kind of more interesting orbit?
Well, so all of theirs are straight lines. So if you look here, all of these should be, I mean, they're probably like a little curved, as you see up here. But there's certainly no like oscillatory behavior or anything. Whereas with ours, at least in our green case, our version of the green case, we have like oscillations going on.
We could definitely do that. We could definitely try plotting the path that they take to this steady state. We have all the data for that, so that's something we could try.
Mm-hmm.
I mean, as I say, I mean, but my background and taste tend to be very dynamical systems. Over the years, I've taken out a lot of that because historically, the students who take this course were not have as much mathematical background, and so the dynamical systems analysis was off-putting to some people. But I think the sort of focus on dynamical systems techniques that everybody is having is great. And the other thing that you could potentially do in this one that could be might be interesting would be, which is one of the nice things about tellurium, is that it's a single line change. You say, instead of simulate, you say Gillespie, and you run the simulation stochastic. And then, of course, you're not going to get beautiful orbits, but you can understand the robustness of the system to noise, and that's a one-line change in your simulation. So it's something that you might, again, that would be more of an extension, but it's something that maybe to think about later on. So certainly, the real systems would be pretty noisy.
Yeah, no, that's definitely a good point. The other thing to note is that like these trajectories are plotted in 3D parameter space, but really there is the dimension of like voltage and calcium concentration too. So like, I'm not positive that you could generate like a flow over this 3D space, really. Like the flow at each one of these points, 3D points, might also be dependent on instantaneous voltage and calcium concentration. And
right. Well, for any given simulation, there will be some orbit. Whether the initial point always gives you the same orbit will depend on those hidden variables.
Right.
Maybe that's why they're seeing that split. In other words, those three populations are because one of the hidden variables is different among the three. Is that why?
I don't think they say anything about that. I suppose it could be true. But that's definitely not where they're drawing their conclusions from. They're saying that it's coming from the manipulation that they're doing, which I don't think is the case. I think it's coming from their initial conditions. But anyway, so that would be our replication phase. And then if that goes well and we're feeling like we have some time, we're thinking that we might go into generating like a rate-based model, meaning that in order for this model to work, you have to arbitrarily specify some target calcium concentration that the system is like trying to maintain itself at, which isn't really biologically realistic. Like there's no little dude inside the cell saying this is your target calcium concentration. So we could develop a model where the rate of insertion into the membrane and degradation of the ion channels is what specifies an equilibrium point, rather than have that be your target rate. I didn't explain that so well, but that would take a little bit of extra work and a lot of research into how these things are typically done because I'm not really familiar with that literature as much. So we could slide that in there or not. And then just app coding. And then, like you said, like data analysis of what kinds of correlation structures we get, maybe adding in some noise, and then doing the write-up.
Well, if you discover that the paper has a mistake in it, you would not be the first person in this class, of the spring class, who discovered that the paper they were replicating was wrong. The degree to which there are minor errors versus major errors is varied, but I would say, I wouldn't say every semester, but I'd say 2 out of 3 semesters, one of the people picks a paper. Usually the ones I pick as in my lists are ones that I know are okay. But when people go outside, it's maybe not surprising how often published papers have mistakes in them of various degrees. I have to say there are papers of mine that are published that have mistakes in them, so I'm not free from this problem. Which is, I guess, the reason that that replication and reproducibility are important even in theory and computation.
Yeah. I'm thinking, I don't think there's any like computational mistakes. I think the mistake was just their failure to consider that the initial conditions that they were picking could have had effects on their results. But I guess we'll see.
I mean, I'm not saying this paper has error. I mean, it's wrong, but people have a couple of times people have found major problems. That simply what they said happened didn't. And again, we accept that in experimental work people make mistakes, but it's a little more surprising when it's theoretical.
So last time we spoke about our topic was based on EGF and ERK model, but I was very much inclined to, like me and my team, we were very much inclined to phosphate involvement in EGF models. So we thought of we read a lot of paper which incorporates the phosphate indulgence in EGF model. So we moved our topic from EGF-ERK model to effects of EGF on active PI transport mediated sodium gene under different conditions. So this is one of the paper we read was based out on intestinal damage, where the epidermal layer of the intestine is recovered using this method, where the sodium-based phosphate is absorbed to cure the lining of the intestine. So our topic has been moved from there to here, and this is same as last. Like, why we chose EGF? As EGF was one of the reason we chose was based on our first motive was to understand the effect in cancer. But then as a lot of paper have been published related to EGF and its relative pathways that are linked to EGF, so we thought of doing something different, because in EGF there is a section where we have diphosphorylation, where it converts the EGF to EGFR and the subsequent pathways goes on. So that was our motive, to incorporate and understand how phosphate includes in EGF signaling pathway. So our timeline is based on, as you can see, timeline is based on, for now we are collecting all the parameters and the data which we need to build our network. Our model is particularly based on MAPK, EGF, and PKA and PKC. So all the three type of signals are being used in order to recover from an intestinal damage. And when there is an involvement of phosphate, all these few of the signal elements are either they give a negative feedback or they give a positive feedback. So our motive is to understand how it works. So currently on the for this week we are working on the parameters and how the what are the compartments we need. So for example, when we swallow food, so there is a pathway that it goes from esophagus to the stomach, then to small intestine, and then to rest of the body. So when it comes to phosphate, it goes a similar way. So if the small intestine lining is damaged, how the phosphate will recover so that the proper consumption of food will be there. And how is it related to kidney as well? Because when there is an enough of phosphate or like overexpression of phosphate, there can be and phosphate toxicity in kidney. So we are now we are now at a stage where we are understanding the biological aspect of how phosphate works and how EGF and the rest of the signaling pathway that I mentioned earlier are used in this. And in the next week we'll be modeling our first milestone, that is implement of EGF and phosphate. And on the subsequent on the next week we are planning to give an analysis and to check whether the expected outcome is there or not, like how phosphate and EGF are ruled. From the second milestone, I'll just go ahead. Yeah. So for the milestone for now, what we have understood is we need two compartments. One is intestine, another one is kidney. So the movement of phosphate, it does happen in two ways. So one is based on kidney and another one is based on small intestine. The graph which is shown in the left hand side is one of the elements we tried to take it from the existing EGF model, where we have taken out only ERK, RAS, and EGF growth factors in both active and inactive state. So all the species is not labeled properly as of now, but we are working on it. So on the right hand side, the image displays all the actual biological pathway, like the process, what it happens when the sodium based phosphate is been used in EGF in normal condition as intestinal condition. This is the motive of our project. So in between in between the milestone 1 and 2, while we are working on modeling the intestinal condition and a healthy intestinal condition and injured intestinal condition, we are trying to model that while as we know that, like as we see, there are a lot of existing models which we have in bio models. We try to take up one of the model and try to add the PI regulation in it, and we're still working on it. We are having some minor issues figuring it out, but hopefully will be done by end of this week or maybe to next week. But yeah, we are working on it. So next goes the milestone 2 and 3. Once we are done with the first compartment of intestine and EGF model, we are planning to implement compartment 2, that is kidney, and the rest of the 3 signaling pathway, that is MAPK, PKA, and PKC. And the final week would be the whole comparison between all the 4 signaling pathway and all the conditions, the healthy condition as well as the injured conditions. So this is what we have planned as of now, and hopefully we'll be able to work on it. Yeah, that's all from our side.
So what happens when you have injury? What is the molecular change or the transporter change that happens during?
it happens like EGF generally inhibits the movement of sodium and PI. So it inhibits and it tries to repair the damaged skin, not skin, the lining of the intestine. So we are trying to model that, like in what condition, like what should be the appropriate phosphate consumption by the human who has a damaged intestine or have a maybe gastritis or something, some of the other intestine related disease. So what is the appropriate phosphate that the person should be consuming? Yeah.
Also, when there's damage, the absorption will be like more. So it will be directly absorbed in the blood and then the levels would go up too quick.
So basically you have normally the epithelium functions as a barrier and the barrier is lost then when you have damage.
Yeah.
So does that mean you need a blood compartment as well?
To be honest, we haven't thought through it. For blood vascular to include in our model.
Well, I'm just trying to understand. You
that's
have
a good
normal
point,
and
mean-
intestinal injury as your examples. Now you said these diagrams are not. I mean, so you've gone from a negative to a positive regulation of the transporter. Is that right?
Yeah. So we haven't thought through the blood flow in the modeling as of now, but that's a good point. I mean, we should have added into our consideration to
Well,
understand.
I'm not saying you need to make things more complicated. It was just a question.
Okay.
Already modeling just the effect in the epithelial lining is probably already a lot. But I was just wondering what the mode of action of the damage response is.
Okay.
Great.
So this is the ideal condition where we don't have a blood flow, but it's only the recovery of the lining, intestinal lining that has to be happened in presence of phosphate. So there should be a adequate amount of phosphate that is required to act as an activator to make the process faster for the recovery of the intestine.
So is phosphate then a critical limiting nutrient for recovery?
It is one of the critical nutrient for recovery. That's what the paper said. And we have read a lot of papers as well that links the recovery of intestinal, the epidermal layer with phosphate concentration.
One of the reasons why we chose phosphate is because in all the biological activities and pathways, phosphorylation is one of the reactions which activate a lot of compounds. So phosphate is important when it comes to signaling pathways.
Well, sure, ATP is important in everything, but the fact that ATP is important doesn't necessarily mean that you're phosphate limited. And I'm not saying it's not. I don't know enough to have an opinion. So just a question. Okay. All right. I guess Peter, did you want to say something today, or do you want to wait?
Yes, I'm ready.
Great. Thank you.
Okay, I'll take a screen share. You'll have to close yours.
Pete, you're not clear enough. Can you speak a little bit louder?
Could you turn off your screen share so that I can put on mine?
Okay, yeah, sure. Thank you.
Okay, I'd like to preface this by saying that I'm in the process of changing my project topic. At first, my topic was focusing on telomeres, Then I couldn't quite find some mathematical models that would be easy to model in NanoHub. And I changed it to more of a immune response or effects of aging on T cells topic. Now I'm circling back a bit, and it's coming back a little bit with telomeres, but here the main focus is on reactive oxygen species. So now I want to develop a focus on proteins. You know, this title is a bit deceptive because it says P21, but there's really a lot that are involved. And my goal is to create an educational app that will guide a user through the roles or the relationships between some of those proteins and how DNA damage response can either lead to senescence or repair. So senescence can happen through several different ways. One of those is short telomeres, but it can also just be DNA damage or some tumor genes. The paper I've chosen for this topic is mainly focusing on a positive feedback loop between the DNA damage response and the reactive oxygen species. And I'll get into that more later. But the main consequence of all this is you'll have mitochondrial dysfunction, and the process of going from some initial damage to actually senescing is a thing that's not too well understood, and it's kind of a long process. So this slide kind of dissects the source paper that I found and will be my primary resource. So their hypothesis is that there's a positive feedback loop between reactive oxygen species production, DNA damage, and DNA damage response. So this is a list of parameters that the paper itself provides, but I may also need some other ones from different resources. So you'll see that it's focusing a lot on specific proteins in the oxygen itself. Then the last one is ionizing radiation, which, if you're unfamiliar, it's commonly used to cause DNA damage response in a testing environment. So the outputs that are given for this paper are kind of already listed in the parameter section. Some of them will affect the others, so that would be, protein abundance and the DNA damage. But I'd also like to focus on senescence and apoptosis itself, which I put a question mark next to it because it has to come through some other paper. It's not through this one. So can we measure the number of cells that die? So that would be a question I might ask. So some things a user might be able to learn from this application are just some basic background about the proteins, what each one does, and what they're involved in. What are the relationships between each one? How do they ultimately contribute to senescence? What are the steps from DNA damage to senescence or actually repairing? What's the minimum amount of reactive oxygen that is needed to cause problems? And is there some threshold where that positive feedback starts to occur and there's a point of no return? And why is that senescence irreversible? So some things I also need to research still, though, are the effects of antioxidants. This was covered just barely in the paper. It's not a natural parameter in their model, but a question I'd like to ask is, why is it that I can't just eat a ton of blueberries and then live forever? Why is it not negate the effects of this model? The second point here is, what should I leave out? I think this is a very important question for me because I got into this. Every time I found a new paper, it seemed like there was a new protein. There was P16, P21, P53, P38, P66. And I learned from studying or looking at someone else's model last semester, or from last semester, I should say, that it's important to have a very specific focus. Don't go too broad. Just get something that gets the point across and teaches someone something useful. Lastly, I might consider looking into some of the genes that produce these proteins, and I think that would be useful as well. So these other two slides just are just showing images of some models that I pulled out of this paper. The point here is not to show anything insightful, but just to show that it works. So right here, I've got DNA damage going up as some of these protein levels or abundances are going up as well. And it's kind of similar, there's the same idea here, where you've got reactive oxygens increasing in blue at the same time that some of the other proteins are. So I'll move on to my timeline now. So the past few weeks have been focused on this section. Although I want to point out my columns first. I've got the task name, the amount of time to complete it, and separately the number of days available, which is a more relaxed guess. So something could only take me 2 days to do, but I'll have it open for several weeks or something like that. And I've also got the when I want to start it and when to end it. So these past few weeks have been focusing on trying to find that new project topic, trying to find some paper lists, which I've covered now. I still need to gain more background knowledge, finish reading the papers, and go into more depth with them. I programmed a very basic test model, which you just saw, nothing too special. I need to focus on what will be my baseline model. When I looked at the past courses work, I saw that you generally want to have some main model that is the starting point for everything else, which you then perturb for the rest of the app. This month, I'd like to focus on parameter scans, and that's this section here. The next task after that is deciding on a second major model. So what I mean by that is, in the tuberculous model, they had a separate model that was focusing on effects of vaccines and another one that was focusing on antibiotics. It did get out of hand a little bit in that sense, but I think it's good to have something that twists the topic a bit, gives you a new perspective on it. If I do go to that route, I want to have some comparative scan as well between the two. The rest of this is adding descriptions to the NanoHub app and doing some of these presentations, these main milestones. The months of November and early December are kind of left ambiguous because of this new change in project topic. And I really want to focus on getting something that's a minimum viable product, so to speak. That's all.
Great. Well, people seem to be very organized this year. I'm impressed. I was going to ask, the one thing I was puzzled by was in the simulation you showed, it looked like the reactive oxygen species come up after P53. And so I would just as a guess have thought that ROS was going to induce P53. And here it seems to be. Is this because you have a model of mitochondrial permeabilization or something like that? I'm just wondering why the ROS are coming up after your P53 signal. I would have, or unless the colors are back.
I can't say that I would be able to explain it too well. I have to read more into the paper, but my guess it has something to do with the positive feedback loop.
But it looks like you're getting the P53 on first. I mean, this is a topic I wish I'd had you around 5-10 years ago when we were doing liver damage. Because one of the problems we had in that work was we knew that the mode of action was elevated ROS levels. And the ROS were causing cell death. But ROS could damage cells in all sorts of ways. They could excite P53, and you could have apoptotic cell death. You could have immediate mitochondrial permeabilization followed by lysis of the cell through necrosis. You could have pyroptosis, although we didn't know about pyroptosis in those days. You could have the damage recovery pathways turned on, and so actually digging into the particular ways in which ROS lead to cell damage or death or recovery is really, to be a fascinating topic, because ROS could damage They could damage the DNA. They could also directly damage the mitochondria. They could damage the endoplasmic reticulum. You could have endoplasmic reticulum stress. You can have protein damage, and then signaling from elevated levels of damaged proteins. And again, I'm not suggesting that you should be trying to include all of that in a model, but just that this question about what's the connection between ROS and cell death or recovery, I think is a very profound one, and so a great topic.
I did just remember these points here are a result of events that are already programmed into the model. But based on what I've talked about, would you recommend any other topics that are related to look into?
Well, I wouldn't. I'm not. You've already got a lot here. So if you could show, even a very abstract way, the feedback between ROS and the DNA damage recovery pathways, and potentially, for example, mitochondrial permeabilization, which releases additional ROS. And so you could show that there are conditions under which you have runaway ROS leading to, in that case, probably necrosis, versus situations in which you have recovery. That already is a very abstract model. Would be quite interesting. Here you're going after much more molecularly detailed work. So I'd be happy to sit down with you and look at the papers, but it looks to me like you've already got a lot. Other comments or questions about it? I mean, one thing that I like here also about this class is we have two groups that are, maybe three groups that are doing very biologically grounded, specific molecular species, very specific damage response pathways, and then people doing very abstract generic network evolution or generic properties of regulation. That variety, I think, is the strength of the. Okay, other comments?
So I was just listening in on the talks, and it sounded like Pete and Connor and Eden, I think, are working together on potentially making educational apps. I was just saying the point that Pete made about having to decide what to exclude from the model is super important, particularly from an educational perspective, because you don't want to overwhelm the user, but you do still want your app to be meaningful educationally. So that's worth, I think, a good bit of time thinking about upfront before you start implementing too much, because that will kind of dictate which pieces of the model you spend your time working on. And then the second comment there I said is that sometimes it's useful to think of a couple of different, because like people, sorry, Joel just sent me a PM about my haircut. I used to have very long hair for people that hadn't seen me before. I went to a wedding this past weekend and decided it was time to get it cut short. So
You were about to say you went to a wedding and they took away your hair.
yeah, it was. They asked for a sacrifice at the altar. So anyway, yeah. So sorry, I lost my train of thought, but
I think you were about to comment on like having parallel task options so you don't get stuck at one point.
right. So I mean, that's maybe a little obvious for a course of this level, but I think what I was more getting at is that for any given educational app, you have to pick which things you're trying to teach. You can't necessarily teach it all at once, again, without overloading the learner. So it may be useful to think about, okay, maybe there's this set of 2 or 3 ideas that I want to teach with this app, but maybe there's an assortment of sets of 2 or 3 ideas that you can teach at once with the app. So it may be useful to think about, if I'm going to teach these 2 or 3 things together, what do I need to include in the model? And if I want to teach these 2 or 3 things together about this phenomenon, what do I need to include in the model? And if you have a couple of different sort of concept sets put together. If you run into issues implementing one, you can switch to another to still get use out of the app, if that makes sense. So I mean, it is a parallel plan thing, but from the lens of what am I trying to teach here.
I think probably the hardest thing for me switching over to modeling was the aspect of like your first point, like really sitting down thinking about what do I need to illustrate this thing. There was a really fun interview with Steve Strogatz and some graduate students a while back where he talked about like ruthlessly simplifying a problem if you're going to do a mathematical model of some kind. And I feel like in biology, when you're trained to think about so many factors at once, it can be really hard to balance those two things. If you're coming from the modeling direction, you have to think about all the things that are interconnected that you're not including that could be relevant to the problem. And if you're going from biology to modeling, sometimes it can be really hard to like get used to representing something in such a minimal way that still captures part of the problem. That's something that I'm still having to get a lot of practice on.
Right. Well, it's a challenge too, because the level of fidelity you need in your model for answering a research question versus teaching a biological concept can be pretty different. So if you're trying to answer a research or build a model that you can use to answer a research question and build an educational tool, you kind of have two different objectives to meet there. Because the people coming to learn about the biological phenomenon may not be interested in all of the details that you need to answer the research question. Yeah, so, I can't give you the answers necessarily, but that's just something to think about, is that you'll have two sets of objectives that you're trying to meet there, as opposed to, trying to find the most elegant single question you can use your app to address. And I'm available by email if people have questions about that. I'm happy to kind of help scope those questions as you move along with your projects, if you'd like. I'll put my email in the chat here. For anybody considering making an educational, sorry, that went straight to Joel. Well, Joel, now you have my email again. Let's see if it does it. Okay, it didn't, it auto corrects my last name to the correct spelling, but my IU email address is incorrect. So I have to always make sure that I'm not sending people the wrong one. But yeah, for anybody considering an educational app, feel free to shoot me an email at some point if you have questions about how to scope that or what you think may be too much for someone coming in trying to learn.
Is your email a single L in the end of your name? That's what you said? Okay.
My last name has two L's. My IU email has one L. No. So it hasn't done me any favors in the constant mispronunciation of my last name either. But anyway, I don't want to take too much more class time.
I'm smiling because I remember I had to pay to get my username for IU. I don't know if you should still do that. In the old days, they'd give you a terrible username and then you could pay them $100 or $150 and then give you a decent.
Yeah, they tell you when you set up your account that you can have them set up mirrors for you, but they don't tell you that you have to pay. So maybe that's a trap.
Yeah. Well, I always assumed they used bad usernames precisely because it was a revenue raising mechanism. They basically shook you down for a decent username. Okay, so I'd like to come back. Thank you. I'd like to come back to what we rushed through a little bit at the end of last time, which was trying to address the question of what are questions we could ask about network dynamics. And these are going to be pretty abstract. And the homework problem, we had a homework problem where we were, people were asked to come up with a problem of interest and then give examples of qualitative, semi-quantitative, and quantitative questions to ask. And people were extremely creative about this. And much more, perhaps much more ambitious than I thought people would be in trying to do it. And so I wanted to go over this again just a little bit to think about maybe some simpler kinds of questions one could ask. And we could maybe discuss those together. Again, Coming back to this idea of a model as something that takes a set of input parameters and model structure, it gives a subset of output metrics. And in the case of network models, those output metrics, at least to begin with, are going to be, because we're in a world of ODEs, these are going to be time series of scalar values. These typically will represent concentrations, but they could represent something else. But we're going to have a bunch of plots which have time on the x-axis and a value on the y-axis. Now we could do more elaborate things that we've seen. We could plot trajectories, orbits, phase space of various kinds. But the base output of a simulation is going to be a set of time series, scalar time series. And those time series will depend on the model structure and the input parameters. And there aren't really, I mean, there at some level an infinite number of questions we could ask about these time series, but they're also at some level classes of questions that we can ask that are relatively restricted. And so we were talking about qualitative questions. And if I have a single time series, so coming back to this idea, so if we look at a single time series, but even if we think about an orbit of multiple time series, fundamentally, qualitatively, there are not that many things that time series can do. We've talked about reaching a steady state. We've had examples of periodic oscillations. We can have chaotic time series. We can have diverging time series, but there really isn't anything else that's possible. If somebody has another example beyond those, that would be fine, but I don't think there really are that many things that can be achieved beyond that. And so then there's a particular kind of question that we can ask, which is for some given model, with a given set of parameters, initial conditions, boundary conditions, does the time series reach a steady state, oscillate, chaotic, or divergent? And fundamentally, those are more or less the limits to the questions that we can ask. If we have perturbations, and in one of the homework problems, we talked about doing perturbations, and I don't know whether we'll have time to get back to that today, then we could go a little bit further, but there isn't that much beyond that we can do. And so ultimately, now the meaning of those qualitative questions, which is I think what people in their homework problems were trying to get at, could be pretty sophisticated. Are cells dividing? Are cells moving? Are cells dying? System recovering homeostasis. But at the level of a time series, there aren't really that many more things that can happen. And as we talked about again last time, but I want to go over it again because we went through it so quickly. The boundaries where you go from, say, steady state to oscillating could be very important. They could be cells going from quiescence to proliferation. They could be a sleep-wake cycle. They could be going from stable populations in an ecosystem to unstable populations. And certainly going from steady state to divergence generally means you're going to die in some sense. And so the boundaries of survivability become quite important. The boundaries of homeostasis become quite important. And so it's of interest then to begin to plot the boundaries of particular regions. And here again, I have this concept of a myofiber, and you have sodium concentration, potassium concentration. You can imagine the muscle fiber sits there and doesn't do anything. If it's a heart myofiber, its normal behavior might be rhythmic contraction, so oscillation. You push it too far, it could become oscillating but in a chaotic way, which could be associated with something like fibrillation. And if you push it really far, the ion channels could lead to the death of the cell. And so you'd like to have some idea of the pattern of boundaries between these domains, both to understand normal behavior and to understand pathologies, and potentially also understand how you might intervene to prevent or recover from some of those pathologies. I think the key thing here is that when you build, as was discussed in the chat, all of these models that we're going to build are radical oversimplifications of biological reality. And so to expect exact replication of results may be challenging. But if we can at least get the relative pattern of behaviors to be right, that's already a lot. So if I know as I increase the sodium concentration in intermediate potassium levels, I go from steady state to oscillation to chaotic. Or if I go at high sodium concentration, I increase potassium levels, I go from steady state to chaotic to divergent. Those patterns might be reasonable to expect to see in an experiment, even if the specific shape of the boundary wasn't quite right. The specific values weren't quite right. And so the identity and topology of the domains in this phase diagram are things that you'd like to get right, even if the details of the model aren't quite right. That's a reasonable thing to say. If you get the topology of the phase diagram right, the model is useful, even if it's not exact. So that was where we stopped last time. I think it's so important, it's worth perhaps going over that again. The second thing I wanted to talk about, and this is one which is in a sense a little bit of a matter of taste, rather than being a definitive question, is what I would call semi-quantitative questions. And the reason that I think semi-quantitative questions are useful to think about is because biologically we very often cannot measure things like chemical concentrations exactly. We have a fluorescent label which is associated with a molecular species, and we know that if it's brighter, we have more of the molecule, but we do not know how much of the molecule a particular level of fluorescence indicates. In fact, in general, because of the way those experiments are done in a microscope, there's no way to calibrate it. And so coming back to this idea of differential measurement, very often we'll say if we make this change, the amount of a chemical increases. We might even know that it increases a little bit or a lot, but we don't know exactly how much it increases. And so semi-quantitative questions are, at least in the terminology that I'm familiar with, are questions that talk about relationships between values and trends, but not values directly. So, for example, to say that the level of a molecule is increasing in time or decreasing in time is a semi-quantitative question. Asking, does an event A happen before or after an event B, is a semi-quantitative question. Very often we want to know about the relationship between stimulus and response. If the concentration of phosphate is increasing, does the level of glucose go up or down? If the reactive oxygen species concentration is going up, does the activity of p53 go up or down? Does mitochondrial permeability go up or down? Sometimes we're talking about levels. Sometimes we'll be talking about slopes, about rates. So if I increase the amount of ATP, does the rate of ion pumping in the cell go up or down? Those would all be examples of semi-quantitative questions. again, the key thing is that we're not talking about the numerical values of something. We're talking about relationships. And those are typically the kinds of experiments that we do. These days, true quantitative experiments are getting easier, but still many things are hard to quantify. Changes are measured. Increases, decreases, rates of change, we can measure, but values not so much. And so that could be done at the molecular level, but it could also be done at a more phenomenological level. Again, is A bounded, infinite, or infinite long time? And you could build phase diagrams. Does the tumor grow or shrink as we increase? First, does the tumor grow or shrink in time? Semi-quantitative question. And then we could say, as we increase the level of a chemotherapeutic, does the tumor grow or shrink? Or does the tumor grow faster or slower? So here, for example, for some putative chemotherapeutic and some anti-angiogenic drug, I could ask the question, if I increase here I've got two phases, the tumor growing and the tumor shrinking. And if I'm trying to treat a cancer, I want the tumor to shrink. So I want to be in that region to the right, which has the blue label. However, I know that both the anti-angiogenic and the chemotherapeutic are toxic to normal cells as well as to cancer cells. And so if I give a little bit, if I don't give it at all, my normal cells are okay. If I give low doses, my normal cells are okay. If I give too much, my normal cells die. And I could ask the question, if I deliver the drug in different ways, does it move the boundaries between tumor grows and tumor shrinks? And does it move the boundaries between normal cells okay and normal cells die? You'd expect that it would. And if I put those two things together, then the region where the tumor dies and the normal cells don't die would be the region of therapeutic efficacy for that combination treatment. And so ideally, what you'd hope is that you have some way of expanding that region of effective treatment. If not, you want to make sure that you are able to deliver it in a very accurate way. And so this is all essentially semi-quantitative work. Now, of course, in the end, you'll be giving the drug at a particular dose. So that'll be quantitative. But even remember with the drug, typically, well, chemotherapeutics are expensive enough and toxic enough that they may actually be titrated by milligrams per kilogram for an individual. But usually, different people with different weights take the same amount of most drugs. They're not adjusted for body weight. And they're certainly not adjusted for things like metabolism, age, and other aspects of individual variability. And so drug dosing tends to be really semi-quantitative. And then we come back to true quantitative questions. We could ask the question, at a given time, is the amount of the molecule bigger or less than some threshold? And the obvious quantitative question is, what is the value at each time? And what is that time constant? But we could ask more limited questions. Is the value above or below a reference level at a particular time? When does an event happen? How fast does the value of something change? What's the slope? How frequently for a periodic event does something happen? And those would be quantitative questions. And so, again, here I'm talking about things that are very generic, they're very abstract in some ways. But the number of questions that we can ask about a time series is fairly limited. And so in the homework problems, when I ask, what do you see? These are the kinds of answers that you might give. How you interpret those things in terms of meaning of the problem that you're trying to address is more complicated. But the number of things that you can actually ask specifically about what you see as output of your data, of your time series, is fairly manageable. Are there any comments or questions about that before we move on? So we can also do differential questions that are quantitative. It's a concentration of A greater than or less than B. That's maybe a little bit semi-quantitative because it's a relationship. Is the rate of increase of A greater than or less than that of B? That could also be semi. So some of these things are a bit, whether you put them in one category or another depends a little bit on how you ask the question. Now, what's the functional relationship between A and B? That's definitely quantitative. In general, we expect that as we go from qualitative questions to purely quantitative questions, our models get less and less good at reproducing reality. We expect the trends are easier to reproduce than exact values. Biologically, most things are stochastic. So even if we had the experimental results and we did the same experiment twice, we might not actually get the same thing twice. But we would expect that there are typical trends that would be reproducible, at least statistically. And so these are the kinds of questions that you might be thinking about in terms of problem analysis. So you already did the homework problem, and I don't know whether we want to take a lot of time on it, but maybe it's worth thinking about it. Let's just take, let's see, it's 5:54. Why don't we take 5 minutes and have people try that homework exercise again, maybe in the light of the problems that you're thinking about now for your projects, and come up just with one example of a qualitative question that you might ask, a semi-quantitative question and a quantitative question that you might ask about your problem. And I used some of the examples we had here. If you increase the strength of feedback between the differentiated cells and the stem cells, does the number of stem cells go up or down? If you increase the level of reactive oxygen species in your cell, does the activity of p53 go up or down? Those might be the kind of questions you could ask. But it doesn't have to only be about levels. As I said, the time at which things happen is also fair game. So if there's an event in your system, whether an event happens before or after something else, when it happens, those are questions that are also reasonable to ask. Levels are not the only thing. Features, temporal events are also reasonable. Any questions about that? Okay, so why don't people let's just take 4 minutes and to see if you can come up with one example of qualitative, semi-quantitative, and quantitative question.
I have a question, James. When we're talking about proportionality, can that also be qualified as quantitative questions? Like if we have a rate, for example, X amount of percent is this type of cell, the other amount is that, can that be considered also quantitative question?
Yeah, well, it's the moment you have a number,
Hmm.
it's quantitative.
Okay.
Now, the place that it's a little bit more ambiguous is, suppose that you measure fluorescence and you say the fluorescence increases 5-fold. That's quantitative with respect to the fluorescence. It's not purely quantitative with respect to the amount of molecule, because you don't know the conversion between the fluorescence level and the molecular concentration.
If there is some relation between intensity and presence of the molecule or something like that, right?
Yeah, it's not always perfectly linear, so it makes it more difficult. On the other hand, if the intensity goes up, you probably have more of the molecule.
Right.
But that's a reasonable question. Again, it's not that there's a hard and fast distinction between these things. But the moment you say, if you say 66 microliters or 5 nanomolars, that's definitely quantitative. If you say bigger than or less than, that's semi-quantitative. So there are certain kinds of words that you associate with different domains.
And proportionality will be quantitative. If I say 1% and other percent, although is, yeah, okay, I got it.
Yeah. On the other hand, if you say a lot more or a little more, that's semi-quantitative. That's why I say there is an absolute boundary between the two. But in some sense, you could think about it as, what is it you have to measure? What kind of measurement do you have to do to be able to answer the question? And again, a lot of it comes about because very often in biology, we can't make the quantitative measurements or fully quantitative. Well, let me call on somebody who does it. Gabriel, do you have an example of something that you could put forward for us about this?
Yeah, I guess, I mean, it's interesting because I was just talking with Connor and Eden, and we were just talking about all the way, like semi-quantitative, I guess when I never, for me, it initially, like the idea that feedback or like bounded versus infinite, all these different things, you know, for me intuitively, I was like, oh, these are quantitative, right? Like, you know, we can, you know, blah, blah, blah. But, you know, actually thinking about it, it's like, oh, okay, yeah, I guess these are not like, it would be much, it's much more, I feel like I have like a stereotype of like how they're usually depicted, but it just doesn't really end up being that way. So yeah, I can give, anyway, just as a caveat, but I can give an example. So I guess for an example of semi-quantitative then would be like the feedback that we see in the Komarova model. So the kind of inhibition or the activation or the positive and negative feedback loops that happen in those circuits, I guess is an example.
So that's a model structure. So now could you reframe that, for example, in a semi-quantitative way, which is to say, if I increase the strength of negative feedback between something and something, then something increases or decreases, or something happens faster or slower. That would be a way of trying to frame that question, perhaps.
Yeah. So if I were to try and think of a different one, if I were to increase the positive feedback between stem cells and transit amplifying cells or the intermediate cells, then that would increase the rate at which, that would increase the rate at which stem cells would turn into transit amplifying cells.
I mean, you could also ask a qualitative question, which is, as I increase or decrease the negative feedback, do I go from a situation which is homeostatic, where the population is bounded, to one where it blows up, or where all the cells die? That would be a qualitative question. And so I think the Komarova paper actually is quite good about defining those kind of. Whenever you have boundaries in a phase diagram, you're basically starting out by asking qualitative questions. When does something change in the way it behaves? Just in a qualitative way. So that's great. Let's see, who else can we grab here? Peter, do you have anything you want to contribute?
I think I would give a wrong answer, but I'll try. So I'm thinking that in the quantitative measurement might be the amount of fluorescence you're looking at. So in the particular paper I looked at, that is a big focus, where they had some imaging. So I'm not sure how that can be measured, if it's maybe like summing up all the intensities or looking at them in particular parts of the model. A qualitative observation would be something like, what's the relationship between some different proteins? And semi-qualitative would be, let's look at one particular protein and see how it changes over time, or what's the abundance.
So I think you're I don't want to say that there's a right or a wrong answer, but I think one thing you might think about, and it was like what I was saying before, is think about the concept of a control parameter and an output. So if you say, what's the relationship between proteins? That isn't an answerable question. You could say, if I change the concentration of protein 1, what happens to the concentration of protein 2? Is a quantitative question. Or if I change the concentration of protein 1, if I increase the concentration of protein 1, does the concentration of protein 2 increase? That would be a semi-quantitative question. Or I'm not sure for a qualitative question on that. Or in my system, does the concentration of protein 1 start increasing before or after that of protein 2? Those would all be possible questions. But I think the key thing is to try to always to come back to the question of what is being controlled and what is being observed and how do they relate. And so the purpose of this exercise, and I realize it feels pretty abstract, and I'm not trying to be critical, is to take a question like, what's the relationship between A and B, which is fundamentally not an answerable question, and to try to come up with something that you could in fact test, a hypothesis that you could test. And so I think the problem that you presented has lots of examples of this, where you could ask that question. And I think some of them were laid out in your slides already. And I remember I asked a question, which is, why does the P53 peak seem to happen before the increase of the reactive oxygen species? So that was trying to frame what I would say here as a semi-quantitative question. Why did I ask that question? Because normally spikes in ROS lead to cell damage, which leads to P53 activity. At least normally maybe is the wrong word, but in the world I know that would be what I would expect. But it's also true that P53 can cause mitochondrial permeabilization and increased ROS. So it could go the other way. So the time order is interesting. There are events in that case. So, and again, these things are slippery because they seem to be too abstract. But when you start applying them to your particular problem, the way you go from a generic topic to a testable scientific hypothesis is to try to ask these kinds of questions. Maybe Danusha, do you have an example that you could give? Are you there, Danusha? Okay. Does anybody else want to? I don't want to cut off the discussion. I have to So somebody else has an example they'd like to present, that would be fine.
Yeah,
Does it,
go ahead, Grant. Go ahead.
I was gonna ask, does it have to be related to our research topic or just a general example?
Could be a general example, anything that illustrates, anything that helps us understand how to frame a question.
Okay, well, maybe you could ask the question of, well, if you somehow block the gene transcription for the production of insulin, then how that could lead to the qualitative symptoms of diabetes in a patient. So that's one way you could go from quantitative to qualitative.
So basically, is there a degree of insulin impairment which leads to a qualitative transition from non-diabetic to pre-diabetic to diabetic?
Mm-hmm.
So yeah, that would be exactly the kind of phase diagram that we were talking about. That would be good.
Can't you get at some of these by looking at the phase diagrams in just like different ways? Like if we have a system where we have like oscillations within one basin, fixed points in another, a qualitative question would be like looking at those dynamics. Generally, semi-quantitative, like thinking about the stimulus response would be something like, if I perturb this much, what response do I get? And that may or may not cross the basin if depending on what the response will be, a change in behavior. And then a quantitative one would be like, you could imagine a case with two fixed points separated, and then you can say, what is the actual change, which I guess that one's a comparison, but you get a concrete value out of it, right? Like if it's a 1D system, you can actually say the difference between concentration is this specific value.
Right, and actually the reason that the homework problems were set up, they were too soon because we hadn't discussed things in class. But the reason that the homework problems were ordered the way they were was specifically because I was hoping that people in that last problem, which I realized was very long, unfairly long, would motivate exactly these kinds of questions. In other words, if you have these classes of questions to ask about the time series that you're seeing, and then you can use those 5 circuits that Herbert has in his homework problem as examples of these different categories of questions. And the reason I asked you to do the perturbations was specifically so that you could check that exact question. So probably if I assign that homework problem this week, it would make more sense than 2 weeks. Anybody else want to weigh in on this? I think it's an important topic, and one that I think, I mean, again, it's discussed probably in some classes, but maybe not discussed as formally as it could be. I think tacitly, we all assume that we know how to ask questions. But in fact, in my experience, learning how to frame questions that are answerable is not easy. Okay. People are free to break in and contribute to that more. Okay, so coming back to this practical thing here, I wanted to talk about events. People seem to have discovered antimony events already on their own, but I asked people in the homework problem to do simulate changes, like adding a drug or a nutrient at a given time. And antimony has a syntax which is name of event, colon, at, parenthesis, logical operator, colon, and then you can have multiple things happen. parameter or variable equals something, expression, comma, parameter variable equals something, comma, semicolon at the end. So if we wanted to add 15 units of concentrate to the concentration of molecular species A at time 15, E1 colon at time greater than 15, A equals A plus 15. Notice that this breaks the use of the equal sign. I talked about how equal signs are only evaluated at the on load. You might imagine this that this is a counterexample. So this equals is different from the other equal signs we talked. There are a couple of things that we need to understand about this. Logically, the statement at time greater than 15, A equals A plus 15 doesn't really make sense, because if this happened literally at every time greater than 15, A would go to infinity immediately. And so we're going to have a concept of a trigger. It's important that we can change parameters or variables. We're going to find that there's certain kinds of things that we can't do. If we have a cyclical definition, it won't work. The trigger could be any logical. So it could be on the value of time, the value of a variable or parameter, or some derived quantity, like whether it's positive or negative. And I need to remind you that there is a bug, may have been fixed in the latest versions of Tellurium, but is not fixed in the NanoHub version, which is if you accidentally specify two events that operate on the same variable or parameter, and they occur at the same time, only one of them will actually act. And the choice of which one is random. And that can be reasonably confusing if most of the time the things happen at different times and it behaves normally, and then they happen at the same time and one of them disappears on you. Those kinds of intermittent errors are hard to debug, so I need to tell you about it. So I thought maybe, I guess I could do a poll for the class. I had a few simple exercises to play with events. If everybody in the class has already got events under their belt, we don't need to do it. If people would like to spend 10 minutes firing up antimony and trying out some events in different situations, we can do that. How do people feel about it?
I haven't tested, but I have a question. We use time there as a parameter, but you could use any other parameter, right? Any other species. If for some reason you say, as this species reach this number, just
Yes.
go back to 0 or something. But that
Right. So that's exactly what my little series of exercises goes to do that.
So I at least am aware, because we already did the last homework, at the general structure of how events work, but I would mind seeing some other examples of how they're used. I think most of the ones in the homework were like Joel was saying, where it's like a trigger event at a time.
Okay. So why don't we do these little exercises? Do you want me to type while you do it, or is it okay for people to just fire up antimony and try it? Why don't people try to implement what I have on the screen here? You don't need all of the labeling, so you can save some typing. I just create an antimony string which says A goes to B at a rate K1A, B goes to nothing at a rate K2B. I'm not sure what the $X0 is. That's probably left over from when I was editing something else. And then try this one and see what you get. Give people a minute or two to do that. Again, it's perfectly fine to type exactly what I typed here, but you can leave out the unnecessary comments and and labels. One thing you do have to be aware of with labels, if you do label reactions or events, those names have to be unique. If I tried to call my event J0, antimony will, tellurium will crash. Another thing that I've mentioned before, which I should say again, is you have to watch out for reserved words in antimony. So time, we know, is a reserved word, has a specific meaning. But the one that often gets people is gamma, because gamma means the gamma function, the mathematical gamma function. And so if you create a variable named gamma, it will crash. So if you get weird crashes in antimony when you're parsing it, one of the possibilities is that a variable or a label was using a reserved word.
What does it
I
actually
haven't.
mean when you named an event? You can call that event for what exactly?
I'm not sure what you'd want to do with it. When you name a reaction, the variable like J0 in the example that's on the screen, J0 is K1 times A. So it creates a variable which is putting the rate, the current value of the rate, which is useful. I don't know what would happen if I tried to
Plot
E1 versus time. My guess is it'll give you the status of the logical. My guess is that E1 would be false, 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 and then true after 15. You could try it out. You could ask if you use that where you ask for the output in the simulate function, where you name the columns you want, you could try asking for output E1 and see what it gives you. If it may crash or it may actually give you a time series. I don't know. Never tried it. What I do know is that you can sometimes do events because the event can be triggered by values and change values. The order of events can matter. So suppose that the first event makes something happen that makes the second event true or not true. Then the order in which I evaluate the events can matter. And so one of the things that you're allowed to do in which we won't cover, but you can look up in the text online help materials, is you can tell it which order to evaluate events in by name. So you can say, look at E1 first, and then E2, and then E3. So you can give them a hierarchy. In general, antimony is designed so that the order of lines doesn't matter. But in this case, it can be dependent.
So, okay, so with the event thing that we have right now, Professor Glazier just said it's like a trigger. So we go past 15 once, it does the step, and then it stops. It doesn't keep adding 15. So
Yeah.
why is it that when we have a sine wave, after it goes over once, it continues to trigger when it meets that definition again?
Because
That's my next slide.
Oh, okay, sorry.
well,
But it's fine, Giuliano. You can answer, and then we'll recover it a second time in a minute.
yeah, so what triggers the event is that expression switching from false to true. And after it is true, it stays true, so it doesn't trigger. But if it goes back to being false and back to true, then it triggers again.
Okay, that makes sense. Thank you.
Okay, so Joel, you can't plot the time value of V1.
Nope.
Thank you for checking. All right. So what did people get something that looked like that? Yes, no?
Yes.
But what you should see is that it increases once and never again. That's the key thing. And so that brings up exactly the question that Connor was asking, which is triggering. And so again, an event triggers once each time the trigger becomes true. So if the trigger becomes true and stays true, like time greater than 15, then it happens once. If the trigger goes false and then true again, then it happens multiple times. So why don't you try that? Why don't you try creating an event at sign of time greater than 0.5, b equals b plus 0.5. Add that to your program. See what happens. You see that the E1 happens once and E3 happens periodically. You may have to adjust how long your simulation runs for, so you see a couple of cycles. Anybody?
Yeah, we basically have a step ladder on mine.
So if I look at my results here, you see the B keeps getting kicked every 2 pi time. That's not a very elegant way to do a clock, but I don't know. I don't know if there's a simpler clock. Okay. So here we were acting on the variable. Now we're going to do something that I would generally say never do in a real problem. One thing people are still having sometimes, I think it takes time to learn how to do it, is translating arrow diagrams into antimony. You want to try to be as clear as possible about the structure of the model. And occasionally people get confused about things like first order decay, or the sign of the direction of an of a reaction. I was going to say ODE, and that's exactly what I didn't want to say, because the direction of a reaction is always clear. The direction to say ODE has a direction doesn't make any sense, and so it's easier to get confused about ODEs than reaction. But let's do something that normally we wouldn't do, which is to say, instead of saying E1 at time greater than 15, A is equal to A plus 15, why don't we make K1, the rate at which A goes to B, go negative? Now, that's breaking my rule, which is that you shouldn't have back reactions except with an arrow. But for the purpose of just learning the method, try it. Tell me what happened.
This was what I got on the left here.
So why don't you run it for a shorter amount of time?
Okay, I will also say that I ended up switching, I changed the problem slightly to one that I had a basin diagram for, so I could look and see how the time events were interacting with that.
Okay. Well, I guess okay, so why don't you change you switched it at time 10? So why don't you just run it, why don't you make the final time 15 instead of 80?
Okay.
So this is another thing that I think some people in the homeworks, something that one has to play with to learn, which is keep an eye out. By default, when we use the plot function, the y-axis is auto-scaled. And keep an eye out for whether the results that you're getting make sense. Chemical concentrations don't go negative, and they don't blow up. And so if you see that there's a little e to 10 to the 45th on your y-axis, it means you made a mistake. Or if the value on your y-axis goes negative, it means you made a mistake. Now, sometimes our models really do blow up. And in that case, what you want to do is adjust the time window so that the values stay bounded in a reasonable range during the time you're looking at. So here, A is
So,
initial.
yeah.
So let's see. I was trying to see. Can we put it back to the one that I originally?
Sure. Yeah.
Just because, not because this isn't right, but just because I think it's a little bit simpler to see it, what's going on with that. Yeah, there you go. That's it. So here you see A decays because A is being converted to B. And then at time 10, B starts being converted back to A, goes back up. Well, that's a good example. I mean, the one Connor did was fine. I'm not saying that it's not a right or a wrong here, but I think that that's a good example. All right. Now, whether that's biologically meaningful or biochemically meaningful, it's not obvious, but it's certainly possible if you're representing something other than chemical concentration that sign could change. Okay. So the other thing is this important comment that you can trigger off of anything, not just off of time. So you could ask the question, is P1 or A less than 1? Or you could ask, is A less than B? So why don't people try that? Why don't you say, instead of at time 10, K1 is minus K1, say if A is less than 2, K1 equals minus K1. Why don't you try that? It's actually not going to look very different. One that might be more interesting would be is if A is less than B. You could try that too. Somebody wants to show me, that would be great. I don't have that. Do people have A less than 2 done? This is what I got. Again, it looks very similar. Is that what people got, more or less? What if you replace it with A less than B? Can somebody try that and tell me what they got?
I did. It's weird. It never triggers.
So A never crosses B?
A never crosses B.
Okay, well then I picked a bad example. Let's see what you got. Let's look at it.
I even put a longer time period to see if ever.
Yeah. So can we, let's make your time period short. Okay, good. And let's see if we can actually pull A below B. So what if we make the decay rate of A fast compared to B? So make, well, you've got K1, make K1 1 and K2 0.2. Other way around. There you go. So now, you're running it, it's running for too long here. So you have to shorten your time. Look at see the values, and the first one of them is going negative, which it shouldn't. And the one is, they're blowing up. So there you go. There you go. So this is also a good example about how to tune the time scale so you can see the thing that's happening that's important. There you go. Is it clear to people what's going on here? A is normally decreasing. When it crosses, we cross B, the sign flips and A will increase. But we now don't have a meaningful kinetics anymore. And so there's nothing to keep A from blowing up because there's no decay of A. So one can play with that. One can play with that rather a lot. And so then the next one, the last one that I wanted to do was suppose that now suppose that you say that K1 equals minus K1 when A is less than 2. And now say K1 equals minus K1 when A is greater than 10. What happens? I said 12, but 10 is fine. As long as it's bigger than 2. So when we write programs, we're used to often, maybe not so much anymore, but we're used to thinking about logical control. If something, for loops, if statements. This is a kind of an if, but it's an if which has some it's event-based programming. I don't know how to say. Otherwise, normally an if statement is executed in line in a sequence of operations. These are things that are floating and happen in the background and trigger whenever a condition occurs. And so in general, we don't know if it's time-based, we know when it happens. But if we have conditionality based on values, we don't know when it's going to happen. And that's actually more the way biology works. There, because as I think Lindsay pointed out, there are no references in biology. There are no absolute times, absolute values.
Isn't this at or this event actually something like an if statement on a continuous loop?
Well, it's implemented that way. In other words, it's implemented that these are checked every time step, micro time step. Okay. And that can give you rise to problems. For example, if you say at A equals 2, because this time stepping of A is done in finite units, it's possible A never has A can go from being less than 2 to bigger than 2 without ever having the value 2.
It will depend how many points you put on the simulate. Yes, it's right.
Right.
It never
So,
reaches true.
so that's something maybe I should have made as a red comment, which is I strongly recommend against using equality in the logicals. Don't trigger on A equals 2 or time equals 6, because it's possible because of the finite time step that you never have that equality satisfied. So I should probably add that to the slide. That you should always use greater than or equal, less than or equal to, never in your conditions. So what happened when you did this one? Anybody?
I received a periodic behavior where A will go up to 10 and stop, come down to 2, and keep looping like that.
Something like that?
That's right. But I'd also like to add to Joel's question about event-based programming. So in this case, I think it's pretty reasonable for NanoHub or Tellurium, I should say, to be constantly checking every time step if a condition is true. But if you're looking to go into this more, you should look into the observer pattern, which is a way to set up triggers in a way that you're not constantly checking, but rather every time a value is set, so you call a function that's like set A, it will look at all the listeners to that event, or the state, I should say, and fire some events.
Oh, there's certainly a lot more sophisticated kinds of event-based programming. This is about the most primitive kind of event that you can imagine. And you're also right that having a watcher that's continually watching isn't necessarily a very computationally efficient way of implementation. But I don't believe you could imagine also, for example, that if an event, in our case, events only trigger when they become true, go from false to true. You could imagine something that checks the argument and has some way of knowing. Well, that's a little bit of a halting problem. That once it's become true, it never becomes false again. Then you wouldn't have to check it. You could save some computing, but this is very naive. The Tellurium implementation is very naive. But your point is an excellent. Okay. Other questions or comments about this? Oh, yes. You you made the Sean made the comment that you can also have logical combinations. So your logical argument could have if time is greater than 5 and sine of time is greater than 0.5, do something. So yes, you can build pretty complicated logical arguments for the event trigger.
And I think it's the language of C, logic language of C, those double E's and then bar. It's, yeah. Antimony is C in the back.
Yeah, it's antimony is Antimony qua antimony is fairly new language, but it's really 98% a language called Jarnac that Herbert wrote 30 some years ago. So it was pre-Python. So it was templated off of C and C++. So that's why you have the exponential, a lot of the comment characters and everything else. See, Terminator character. Okay. So again, I think this was, people seem to manage this just fine in the homework, but I thought that the intention was that these two comments were, these two exercises that we just did would have been before the homework that you turned in last week. On the other hand, having tried those homeworks, I guess at least maybe you appreciate more why we're doing these things. All right. Any other questions or comments about events and triggering? Again, there is more that can be done, and you can look in the documentation if you want. The one thing I'll warn you about again is that if you have two events, if you have two events that trigger at the same time and they act on different variables, different parameters, they're fine. It only fails if you act on the same thing at the same time. And so if you say at 3 times sine time greater than 0.5, A equals A plus 4. At 5 times time greater than sine of 5 times 5 time greater than 0.5, A equals A minus 4. That will work whenever the times are not don't have a common divisor, but at time 15, when it's 3 and 5, only one of them will act. If somebody wants to try that out, see what happens. So at time, say at sine, at 3 times, sorry, at sine of 3 times time greater than 0.5, A equals A plus 1. At sine of 5 times time greater than 0.5, A equals A minus 1. Can somebody run that and see what happens? See what happens at time 15. At time 15, it should be 0 because plus 1 and minus 1 should cancel. But I'm pretty sure you'll see it spike one way or the other. That was a bug this class caught. And I know Herbert said they were going to fix it in the next release of Tellurium, but it hasn't been put up on that, I don't think. So if you have a desktop version of Tellurium, a new one, it should work, should be correct. But on NanoHub, it should break. I wanted to say, I guess it was Lindsay, you were running Tellurium in Colab, right?
Yeah, I did pip install.
Yeah, so that's the thing. Colab is wonderful, and in many ways it's more enjoyable. You know, it's a nicer experience than NanoHub. But I was intimidated by having to have people have to do a pip. How was the install? Was it a pain or was it okay?
I mean, I have to run it every time I open the notebook, so that's not ideal, but it only takes like 40 seconds or something. Not too crazy.
Maybe that's another place where I should ask you to give me one slide on how to do it. And next year I can add that to the deck so if people want to try. Because Colab is much more modern than that. But you do have the problem that it's not an installed package, pre-installed. I meant to say something when you showed us that. And thank you for doing that.
It was Grant's idea.
Okay, thank you, Grant, for doing it.
In all fairness.
Well, I'd wanted somebody to try it out on Colab, so you got it done without my having to ask. So what happened when you tried to do the temporal interference on the events? Did somebody get it? Try it?
I tried it. It crashed somewhere here. I don't know if I even listened to all the instruction you gave, so I'm sorry.
You wanted to show it?
Sure.
But I don't have Tellurium up. So. Actually, I do, but I don't have everything tight.
I don't know if that was the thing you said. Okay,
Oh,
want me to...
because we've got the names of the events the same, right? Okay, so no, so at sign time greater than times 5, greater than 15. Don't combine them. If you combine them, it'll work.
Oh, okay.
Just make it 2 lines. E1 at sign
Oh,
time,
okay. Mm-hmm.
right, times 5 greater than 0.5. And then you need a column. There we go. And make it +1 instead of 15. Okay. Okay. And now the same thing, new name, new variable name. And say now make it times 7. 7. 3 is fine. Okay, 3 is fine. 7 minus 1. Now let's run that.
Wait, I have some error.
Get rid of all the junk.
Hold on a second then. Where?
Mismatched parentheses.
Line 10. Is it because I don't have this here? Does that make sense?
Your parentheses aren't matched.
Oh. Yep. There we go.
Okay, so the problem here is which one is the one that's going to cut, which is the one, it's time 15. Yeah, look at time 15. Nothing should happen at 15.
Oh, yes, it's turned off. Yeah, okay.
But it spikes. So it does it picks one. And I think it's picks random. I remember that. It may pick the first one, but I think it picks random. All right, well, I don't want to spend too much time on that, but we did a lot of tail chasing over that bug, I have to say. But that was not a bug that I expected. In other words, Tellurium is generally quite a well tested package. And it has a lot of users, and so finding a bug in it was a bit of a surprise. All right. I think what I want to do instead is here is jump forward. And I'd like to spend, since we only have about 15 minutes left, I would like to jump forward to to just work on some homework problems. And again, a lot of people had no issues at all with the homework. But I wanted to come back to because the feed forward network is rather I don't think I can get through a meaningful amount of it in 15 minutes. So do people remember this homework problem? Yes, no. As I say, it was a bit long. I should really have broken it up into pieces. So in this problem, Herbert gave us 5 simple circuits, a negative feedback, positive feedback, 2 components with negative feedback, 2 components with positive feedback, and 2 components combining negative and positive feedback. Is the screen share working?
Yeah, we can see it.
Because I just got a funny message from Zoom on my screen. Okay. And I had asked you to go through in each one of these. And first, and I realized that sometimes there are a lot of steps, but the steps are designed to be not so much ends in themselves, but ways of thinking about a problem. First, display the time series of A and B. And there's this important point, which is you need to learn to adjust, and we did this just now. You need to adjust the time window so that you see the thing that's important. And you may have to change that time window depending on the parameter values of the question you address. And then doing a parameter scan over initial values of A and B. For cases A, the first 2 cases, there's only A, so there's only 1 parameter. In the other case, there are 2. And ask the question, what are the steady state values that you reach? And in particular, the key question is there 1 or more than 1 steady state value? And of course, for people who do dynamical systems, this is a fundamental question. Bifurcations are changes of the presence of fixed points. And so as you change parameters, the appearance and disappearance of fixed points is the critical question for bifurcation analysis. And then the third question was what happens now when you change. So the second one is what are the fixed points? And that's a question for a fixed set of parameters. And then number 3 is a bifurcation analysis. As you change one of the parameters in the system, what happens to the fixed points? How do they change? And then number 4 was asking the question, if you perturb the system, does it return to the initial fixed point, or does it jump to the other fixed point, or does it do something else like diverge? And so the sequence of questions that were asked here was precisely what you do when you do a dynamical system analysis of any system. So the people coming from Randy Beer's class will recognize this then as sort of the standard logic about the way you think about analyzing a problem. Since we haven't really looked at fixed points and stability formally within the class, I didn't frame it in that way in the assignment. That's where these questions are coming. So maybe it's worth doing one of these together in our last few minutes. So the first problem was autoinhibition. And if people are very sophisticated and they've looked at self-inhibition, they might expect that autoinhibition leads to oscillations. But it winds up autoinhibition only leads to oscillations if you have delays. And so by itself, this autoinhibitory network leads simply to a single fixed point. It's not a very exciting problem. There's a single stable fixed point, and you can plot the value of the fixed point as a function of K1. And the issue about sensitivity was how tightly regulated, how quickly do you come back to your system, and how sensitive are you to changes in K1. But that one isn't, I don't think that one is one we want to do right now. And so I guess I would like to work with you on this one. And again, when I say work on it, maybe I'm asking you to do it. A lot of people got this one fine. A number of people in the class had some challenges with this problem. A reasonable number of people had trouble translating the arrow diagram into antimony. And I know that takes practice, but it's critical that you learn how to do that. And so I want to make sure that people can do that. This one apparently seems very simple. A activates A. Easy. But it has moderately interesting behavior. And so maybe in our last 5 minutes or so, I'd like people to implement this one. If you already have it from doing the homework problem, that's fine too. But I'd like if somebody can show me, especially number 2, which is as I change my initial values of A and B, what happens to my final values? That's the question: is there one stable fixed point, or 2 stable fixed points, or something else for this problem?
Professor Glazier, for this one in the homework on top of the tellurium, I did generate a phase portrait. I don't know if that would be helpful for what you're trying to get at here, but if you want it, I have it alongside the tellurium stuff.
So your phase portrait is for different initial values of A and B?
So I have it with the 2 equilibrium points and the unstable one that has the base and boundary, so you can see how the initial condition impacts it.
Sure. I mean, why don't people work on this on their own, and then I'll have you show that nice the phase plot. I'll give you the screen share. If I were being a little more dynamical systemsy, I would show people how to do the phase portraits, and then have them do exactly that. So again, if you did you do it in antimony, or did you do it in Mathematica or XPP or something else?
So these ones I did in Mathematica. They're just because I've tried doing it in Python before. The workflow in Dynamic and Mathematica is a bit faster and more smooth, but you can generate the same thing in Python.
Yeah, it's not bad. You basically do a 2-parameter scan for initial values of A and B, and then you plot the orbits. If you want to put the arrows on it, it's a little more work. But yeah, do you want to show us the Mathematica result? People should try this on their own. Be trying it in antimony while he's pulling this.
Can you see the Word document on the right side and tellurium on the left?
Yeah.
Yeah.
Okay. Unless I made a Mathematica error, I think this corresponds. So I have I have 2 equilibrium points and an unstable one. And then here on the left, I did a perturbation from the lower one past this unstable equilibrium point and show that it goes to where the other stable one is and equilibrates. And then down here, I show that if I push it up, but it doesn't reach the value where it crosses the basin, then it will go back down to the original equilibrium point.
So boy, I have a whole slide deck going through exactly this analysis, which I didn't show this here. Maybe it's just worth for people who are not dynamical systems, people just explain what the blue and the red fixed point notation on your line.
So the
dynamical systems class.
So generally, each point on this line is a fixed point, meaning that the rate of change is 0 at this point. So if you are on exactly any of these 3 points, the rate of change is going to be 0. So that's how we get where those are. That's what we've been doing in terms of the analytical solutions in general. How you then end up getting the stability is in terms of what happens when you're slightly off this point. So that has to do with your derivative at that point. So basically what blue and red mean, blue is an attractor and red is a repeller. So that's what these arrows are trying to show here. So what you would see is, if you just played with it, is that if you push this thing over here, right, it is going to end up going back that way. If you push it towards 0, it's going to come back to this equilibrium point. That's why for this one, when I increased the value of A, it ultimately ended up going back to that lower equilibrium value. There's this, if you have smooth and continuous equations, your trajectories have to be consistent. So we need something, you can't all of a sudden go from headed in this direction to this one without a point where the rate of change is going to push you. So this is what separates what's called the basin of attraction. It's like the orbit within the states where you will end up at one location or another. And for 1D, you can solve this also by plotting the function that you're dealing with and looking at the slope. That's probably the easiest way to visualize it, but I don't have a diagram for that.
Okay, so you've got 3 fixed points, two stable fixed points, the blue ones, and an unstable fixed point, which is the red one. And for thinking about an unstable fixed point, think about balancing a pencil on your finger. So if you balance it exactly, it's there, but if you push it a little bit either way, it'll fall over. So think about that as an unstable fixed point. So if you're exactly at the bread point, you stay where you are. If you move a little bit away, you either go down or up. Now here, the last thing I asked you to do the perturbation, which you've done. The other thing to think about would be the bifurcation analysis. What happens to the positions of those fixed points as you change your parameters?
So I think that for this one, I actually did do a bifurcation diagram. I didn't do this for all of them, but yeah.
So this is in N.
Yeah, so sorry, N is what I used for H. That was just
right.
So basically, this goes from having, when you're at this parameter value, you have a single equilibrium point. Once you get to, what is that, like 2.4-ish, you have a full bifurcation, and then you're going to find 3 equilibrium points, which is closer to the, which is in the same family of phase diagrams that we just looked at.
Right. So I wanted to talk about, if we go back to the diagram, just the 3-point diagram for a second.
That was, that's this one, right? No, here we go.
There we go. And now we look at our parameters. You're changing the exponent, which is interesting, but harder to understand. If I change the decay rate of A, that is KD, if I make KD bigger, what happens to the values of A? What happens to the equilibrium values of A? Do they go up or down? If I make the decay rate bigger.
It's going to go down.
Goes down. If I make the decay rate for A too big, what happens?
I mean, you should get a point, right, where you also get a bifurcation. Everything's going to trend towards zero if the rate of decay is too high.
Yeah, that's right. And so Connor talked about this a little bit. Fixed points appear in pairs and disappear in pairs. You can't create them by themselves. So you create a stable and unstable fixed point as a pair, or when a stable and unstable fixed point collide, they annihilate. And so if you increase the value of KD, what you'll see is that the value of that upper fixed point, that blue one, will move to the left. And eventually it'll clobber the red one and disappear. And you'll just get, the only thing you'll get is 0, or a very low value of A. You'll have nothing. You'll get actually B over A. B over KD will be the only fixed point. Similarly, and this one you can do analytically, which is why I asked you to do the analytic. Similarly, if I increase the value B, which is the rate at which I pour A into the system from the outside, that pushes up that lower fixed point, that left-hand blue one. And if I make it big enough, the blue and the red will bump into each other and disappear, and I'll be left with only the high value. And so I can actually take the system, which normally has low and high stable states, and if I push it too hard in the decay side, I abolish the high stable state. If I push it too hard in the dry side, I abolish the low stable state. And so actually, if you scan those two parameters, you get non-trivial bifurcation. So for people who've never seen dynamical systems, that may seem maybe a little bit of a harsh introduction. But
sorry.
It's something to, nothing, I asked you to do it and I appreciate it. Again, given the interest people have in dynamical systems in the class, I thought it might be nice to talk about things in that language a little bit. And Herbert's textbook does have some chapters, later chapters which do a dynamical systems analysis. It's something that we can go over. Actually, I have in my slide decks for the toggle, I do the bifurcation. Okay, anything, we're a little bit over time. Is there anything else people want to talk about now before we break? The other homework problem I really wanted to do was that number C in that homework problem. And I'll just remind you of which one that is. And maybe somebody would like to present that for next week if they feel like it, which is this relaxation oscillator. And some people got this right away. Some people had trouble with it. It's a subtle one because what you get depends on your initial conditions. For some parameters and initial conditions, you get a stable steady state. For others, you get a periodic oscillation. And this is pretty interesting because when you hit the thing with the pulse, you can switch it from stable steady state to oscillating. And you can also switch it back. You can turn it from an oscillator back to a stable steady state. And so this one, it was a little bit of a cheat to say, find the steady states, because sometimes it's an oscillator, not a steady state. But that of the circuits that Herbert shows, that's the one that's the most interesting. And it's the one that also takes a little bit more effort to analyze. I know some people found the oscillator. I don't know whether anybody actually did the phase diagram for the oscillator.
I have one, but it's very ugly, and it was the one that I was less confident in.
It's a little tricky because finding steady states is easy. Finding the Hopf bifurcations for oscillators takes a little bit more work.